All right, welcome everyone to the 2022 PI Conference. My name is Christopher Scal-Chunes and I'm Vice President of Research at IDF. I'll be the moderator for this education event. Today, Dr. Emily Ricotta will be presenting the importance of clinical trials. There'll be time for questions at the end of Dr. Ricotta's presentation. Please submit any questions you have to the presenter in the Q&A section on the platform. I'll be going through those and we'll answer those at the end. So with that being said, I'd like to welcome Dr. Ricotta. Dr. Ricotta, please, the floor is yours.
Thanks so much. Hi, everybody. I'm really glad to be here today. Thanks for the opportunity to talk to you all about the importance of clinical studies. I've got to give a quick disclaimer. All of the opinions are mine and do not necessarily reflect those of any of my employers or the federal government. So let's talk about clinical studies. A clinical study is one that involves research using human volunteers that specifically adds to medical knowledge. There are two main types of clinical studies. They're going to be clinical trials and observational studies. The difference between the two is that in clinical trials, the exposure is controlled by the research team. In observational studies, we don't give anything to any of the study participants. We just watch and see what happens. Why do we do clinical studies? There are a number of reasons that we may run a clinical trial where we're actually researching using human subjects. This could be to evaluate different interventions, such as new drug therapies, different types of procedures for treating a particular disease or condition. We might want to find ways to prevent disease development or recurrence, such as vaccines or some sort of lifestyle changes like exercise programs or diets. We need to evaluate diagnostics before we put them out onto the market to make sure that they are, in fact, detecting what we think they're detecting. And we can also use clinical studies to discover new risk factors for diseases. There's a whole lot more about what clinical trials and clinical research is on the clinicaltrials.gov website, which I have linked here. So check that out if you want to hear much more detail than what I'm going to be able to give you today. So when we are going to do a clinical study, the first thing that you have to do is develop the research question and the study hypothesis. Even exploratory studies need to have some sort of general idea what you're going to be looking for, and having a defined study question helps select the appropriate study population, choose the correct study design, and make sure that we are measuring the correct outcomes and other factors that could be important. So, for example, a research question might be, do people with immune disorders develop an antibody response to COVID-19 vaccines? This differs slightly from the hypothesis, which in this example could be most people with immune disorders will develop an antibody response similar to healthy controls. So sometimes you'll have both the research question and a study hypothesis. Sometimes you don't have a hypothesis at all, and you're just trying to evaluate the question because you don't have any preconceived data or any idea to help you develop a hypothesis. Next, and a question that is really relevant for maybe some of the listeners today, is how do we select our study participants? Who gets to be included in a study? One of the main questions, as an infectious disease epidemiologist, I would ask is, how generalizable do we want our study results to be? By this, I mean, who are these results going to apply to? So if I go back to my study question, if I want to assess the immune response to COVID vaccines in people with immune disorders, and let's say I only select children who have antibody deficiencies, the results of my study are unlikely to be super applicable to people with other immune disorders, and they might not even be really relevant to people who have antibody disorders but are older, because we know that antibody production decreases with age. So these are the kinds of things that we need to think about when we are enrolling people in our study to make sure that the results are as widely applicable as we want them to be. We also need to think about the ethics of recruiting people in studies. Frequently, this is going to apply to minors and prisoners who may not be able to provide truly informed consent to participate in a study. It could also apply to pregnant people who we specifically want to keep safe, especially if we're going to be giving any sort of investigational treatments. There are a number of different things that we need to consider when we talk about the ethics of enrolling people in clinical studies. And so it's definitely something that needs to be considered upfront. Finally, logistics can dictate who's actually going to be enrolled in a study. For example, if I need to do a biopsy in my study and I need to bring people to the NIH to do it, It's logistically simpler to bring people who live around the National Institutes of Health to come in rather than flying people around the country to do so. In some cases, that is warranted and we can do it, and so it's not a logistical barrier. In other cases, it is. So sometimes logistics are just going to limit who we can or cannot enroll in a study. So one of the main thing that I'm going to really talk about today are the study designs. And the study design that you choose depends upon your scientific question that you're trying to answer. Not all study designs are created equal in terms of the quality of research evidence that they can provide. So case reports at the lower part of this, the research pyramid here, those are going to be studies that just have maybe one or two people or a couple of people reporting on something that happens in this group, and they're not very generalizable. So, if we see something in one or two people, that's unlikely to apply to the entire population. So we can't really rely on those as much. This varies from randomized clinical trials, which is the top of the research group of quality evidence, that are considered the gold standard, especially when we are trying to evaluate treatments or interventions in clinical trials. I'm going to start with talking about non-randomized study designs. So non-randomized studies are the studies in which participants are not assigned by chance to the different exposure or treatment groups. This could be because the participants get to choose what group they want to be in if they decide that they want to try a particular medication or not. They may be assigned to groups by the researcher in a fashion that's not random and not unblinded, or not blinded, excuse me. They could possibly end up in groups by some sort of inherent characteristic that they have, such as particular immune disorders. One of the big non-randomized study designs that I'm sure you've heard about are cross-sectional studies. These are studies where we look at an outcome in a group of people at a particular point in time. So for example, we might get a group of people and test them once to see whether they have COVID antibodies. We don't care about whether they had them earlier. We don't care about whether they have them later. We just want to know right now how many people in this group have COVID antibodies. The nice thing about these studies is that they're very quick and easy to do, and they don't require follow-up with participants. So if people move around or people decide they don't want to participate anymore, it doesn't matter because you only have to measure them once. With cross-sectional studies, though, you can't determine causality, which is whether A causes B, because we don't gather any information on what exposures may have occurred before that snapshot in time that we're taking. It can also be hard to study rare diseases or outcomes in cross-sectional studies because there just isn't enough time for a large enough group of people to develop the disease or the outcome. So instead, maybe we would choose a cohort study. These are longitudinal studies, and by that I mean we follow participants over a period of time. This can be from days to weeks up to years. In cohort studies, the participants are selected up front for having particular characteristics, such as having a particular immune disorder or having been exposed to COVID-19. And then we follow them over time to see whether our outcome of interest occurs. With these studies, we have a better chance of assessing causality because we are choosing a spot in time where they don't have our outcome, and we know that, and then we follow them through to see if they do. And so we have a better chance to see, okay, well, maybe A causes B. In the example that I have on this slide, we could study whether viral infection is associated with an increase in heart attacks. Now, the disadvantages to cohort studies are that you usually have to follow a large number of people or follow them over a long period of time to observe enough instances of your outcome to make any sort of statistical conclusions. This can be logistically difficult for both the researchers and the participants who may have to submit samples multiple times or go to the study site repeatedly. They can also be really expensive studies. The nice thing about them is that we can do both prospective cohort studies, which means we pick our people and then follow them forward in time, or we can do something called a retrospective study, which is frequently when we look in medical records or we look in registry data, and we select our participants based off of an exposure of interest that happened in the past, and then we look forward in time through their records to see whether they have an exposure. The last non-randomized study design I'm going to talk about today is the case control study. So this is the inverse of the cohort study. In that other one, we selected our participants first based off of characteristics that they had and then watched for the outcome. In case control studies, we select people based on the outcome, and then we look backward in time to see whether they had had any particular exposures that we're interested in. There are a number of other non-randomized study designs, such as experimental care, surveys. I'm sure you've all filled out a survey before. That was a non-randomized study, focus group discussions. So they're varied, they're many, they all serve different purposes. I won't go into those in detail, but I just want you to know that they do exist. So what are the pros and cons of non-randomized studies? Well, they're generally easier and cheaper than randomized trials are. They don't require such rigorous methods usually to develop, which has its own pros and cons. I won't talk about that today. But these can be faster to implement and complete than randomized trials as well. Again, you don't have to accrue a certain number of participants necessarily. You don't have to go through quite as much rigorous regulatory approvals or things like that. They can also be easier to run across multiple settings. So if I want to study an outcome at a bunch of hospitals in the US or in a bunch of different countries, non-randomized studies can be easier to do. They can, however, be difficult in terms of identifying the cohort. So you can't just pull anybody off the street for a non-randomized study. You really need to make sure that you are selectively including your participants. The endpoint can take time to occur, which I mentioned. It can be difficult to establish causality because of external factors that may influence our result in ways that we didn't expect. They're also really easy to do badly. There's unfortunately a lot of evidence out there of non-randomized studies providing us really not great evidence for things like treatments or interventions, and then we've run with them. And that can do a lot more harm than good. So now let's talk about randomized study designs. These are studies in which participants are assigned to study groups totally by chance. When you think of clinical trials, you usually think of a randomized clinical trial. So why do we randomize? Well, randomization helps prevent bias. Bias is any error in a study that can cause a false conclusion. Bias can be introduced intentionally or unintentionally. This occurs when study results are affected, again, by factors not related to the treatment being tested. And so we are trying to understand the relationship of our exposure of interest, let's say a drug and our outcome, and other factors are going to potentially influence either people getting that treatment or experiencing an outcome, and then we can't necessarily tell what our result is going to be. However, randomization aims to actually take that out because we're going to be putting people randomly in one group or another, and just with the laws of large numbers, we should just be randomly putting, the same number of people over here as in the other group. So let me give you an example. So let's say we want to study whether drinking coffee causes heart disease. We're going to look at people who drink 1 or more coffees a day, and we're going to compare those to people who do not drink coffee. Now, since we don't randomize our study, we end up with more smokers in the coffee drinking group than the no coffee drinking group, just because smoking and coffee goes together. When we do the analysis, we see more heart disease in the people who drink coffee than those who don't drink coffee. And so we conclude, well, drinking coffee causes heart disease. However, we have confounding by smoking because we know that smoking causes heart disease. And if we had randomized to you people in group A drink coffee and people in group B don't drink coffee, we probably have ended up with a similar number of smokers in both groups, and therefore wouldn't have seen that association between smoking and heart disease, and likely wouldn't have seen an association between coffee and heart disease. So there are multiple ways that we can randomize. I'll cover a couple here. This is a parallel trial, and in parallel group randomization, we assign participants randomly to receive a certain treatment, and then each person stays on that treatment for the duration of the study. This can be really useful for a lot of different diseases. It allows us to run certain experiments simultaneously in different groups. These groups can be all over the place. It's a really functional, really useful design. We can also do something called a crossover trial. So this is when participants receive a sequence of different treatments. So they could start off receiving a treatment, the new treatment that we're experimenting on. And then after a certain amount of time, they stop getting the new treatment and they then receive a different treatment or they receive a placebo. So the advantage of this is that every person in the study acts as their own control. And so you don't have much external variance happening because of those confounding factors, because they're controlling for themselves. You do, however, have to have a washout treatment between the studies. And so if you're receiving the new experimental drug, you have to wait a certain amount of time for all of that drug's effects to leave your body. And that could potentially leave people untreated when they need to be receiving some treatment. And so these aren't appropriate in all cases, but they can be very useful if having that washout period won't affect the illness of the person being studied. Finally, we have something called a multi-arm, multi-stage design. This is what we would call an adaptive trial design, and these are all the rage. These study designs provide the option to modify the study as the trial goes forward. So what happens is that we will start our study with a certain number of treatment groups that we'll be testing, and then we'll do an interim analysis. So before the study is actually complete, we'll stop, we'll analyze the data. This allows us, if we see certain treatments that just aren't working as well, or maybe some treatments are working really well, we can stop those treatments that aren't working and move those participants to a different arm of the study. Which is really nice. It saves money, it saves time, it helps participants potentially receive more active therapy. And it is, like I said, it's a newer way, a newer study design that's really being implemented a lot because it gives a lot more flexibility in what kinds of things, how many different things we can try all at once. So there are, just a note, there are a number of other different ways that you can randomize things. They get more complicated from there. There are actually people who study specifically the epidemiology and the statistics of randomized clinical trials because they are so complicated. So I'm not going to go into more detail. If you're interested, again, you can check out clinicaltrials.gov. So when we're doing these studies, there are a number of different ways that we can be comparing our new drug or our intervention. We can compare our intervention to a control to see if this new drug is better than whatever the control is. So this is if A is going to be better than B. That's a superiority trial. If we're interested in just seeing whether the new intervention that we're trying, the new drug, is no worse or no better than the control, that's called an equivalence trial. So we just want to make sure that whatever it is we're giving to somebody is the exact same as what we've been giving them before. Finally, we could do something called non-inferiority. These trials are when we are trying to determine if the intervention is at least as good as or possibly better than whatever the control drug is. So new drugs, we hope, would be better than the older drugs for whatever reason. You know, they've got a better mechanism of action, or they make the person taking it less sick. But at the very least, they need to be as good as what the previous standard of care has been, or nothing at all. So that's what these non-inferiority trials are. So again, a number of pros and cons to this study type. Pros of randomized control trials are that you can control bias through randomization, like I said. So if you want to make sure that you don't have any sort of confounding variables causing an association between your exposure and your outcome, then randomization should hopefully get rid of a lot of that. You can establish causality, which is really what we're looking to do. We really want to make sure that our exposure is causing or preventing our outcome. So that's why randomized control trials are really the gold standard if we're trying to demonstrate causality between an exposure and an outcome. The results are really easy to analyze with well-known statistical tools. When I say easy, I mean they're really difficult to analyze, but we can use very straightforward, very well-known statistical tools. It is complicated analysis usually. And the populations of participating individuals are clearly identified. You know who got your exposure, you know who did not get your exposure because they were assigned that way. These are expensive in terms of time and money to run, though. They can be very difficult to set up. They can be very expensive. You really have to pay attention to the study design, and then you have to make sure that your participants stay on the treatment that they're supposed to, or you really have to be careful in your analysis accounting for that. Loss of follow-up can be a problem when it comes to clinical trials. If people just drop out of the study for whatever reason, that has to be taken into consideration. And they can be difficult to implement across settings, unlike non-randomized studies, which are a bit easier to do. So to summarize this section, I know I went over a lot, but really, the important points are that clinical studies should always start with a research question. You're always going to want to clearly define the thing that it is you're trying to study. You really need to choose the study population wisely to make sure that the results of the study you get are going to apply to the people that you want them to apply to, because not every study is going to apply to everybody out there. And then the study design depends on that research question. There are so many different study types that you can choose from. It's really dependent on your research question, on your study situation, on the logistics that you have. And so you really want to work with a multidisciplinary team in order to figure out what the best options are going to be for your study. But there are a number of different ways that we can get at a scientific question and evaluate it. And we have so many people who are experts on all of these different things. So it's a really clinical trials are a really rich way to get at information and improve population health. All right, I'm going to give you the fastest of overviews of the study that we are running at NIAID. We are looking at the immune response to COVID vaccines in people with immune disorders. The objectives, the ones that I'm specifically going to talk about today, are to characterize the antibody response in people with different immune disorders. We're looking at vaccine-associated adverse events, which I'm actually not going to cover because they're very safe vaccines. Excellent, yay. We're also trying to assess the incidence and genetic sequence of post-vaccination SARS-CoV-2 infection. We have a number of other objectives and things that we're going to be looking at as well, but I'm not covering those today. So ask me about them later. This slide, there's a whole lot going on here, but when we start to look at antibody response, what we see is that after 3 doses of the vaccine, the antibody response dramatically increases across the board. So while the antibody level doesn't necessarily get as high as that of healthy volunteers, we do see a really big increase, which really demonstrates the importance of getting a booster dose, at least a 3rd dose, if not more doses. We also see that after dose 3, the antibodies actually are more durable. And so they stick around longer than they do after dose 2. So after 6 months after you receive your 2nd dose of antibodies, we see a very sharp decline. We see it in healthy volunteers too, but in everybody it goes way down. After your 3rd dose, your antibodies don't go down as quickly and they don't go down as far. So that's another reason to really get a booster dose and probably continued doses. This is something that we're going to continue looking at after 4 doses, 5 doses, this new booster vaccine. So keep your eye out for more information on that. I did also want to highlight quickly for folks that received Evusheld, this was FDA authorized during our study after most people had already gotten a 3rd dose of the vaccine. So you can see in people who got Evusheld, that's that orange group along the bottom, they were primarily non-responders. They didn't produce any antibody to the vaccine. But once they got Evusheld, their antibodies went up, which is really great news. So if you are an individual that is worried that you haven't had an antibody response to the vaccinations, you may want to check with your primary care providers whether Evusheld is an option for you, because it does look like it will provide some level of antibody protection. The next thing we checked for was correlation. So what we wanted to see was whether the amount of antibodies that people were producing correlated with how well that blocked the virus's ability to actually bind to the cell surface. And so what we're looking at here is a graph. On the x-axis, we see the amount of antibody that's being produced, and on the y-axis, you see the amount of inhibition. So how much virus is kept from binding to the cell receptor. And you can see the thick orange line is overall, there's a relatively decent correlation. The more antibodies you produce, the more virus it's going to inhibit. Great. But we made these vaccines using the original Wuhan strain of the virus. So the mRNA that you had received in the earlier set of vaccines, this is all based on the Wuhan strain. And so predictably, when we look at the correlation between binding and inhibition, we see that it is higher among the Wuhan variants. And then as we progress through the different variants, we see that correlations start dropping. So even though you may have a lot of antibody, that antibody is just not doing as good of a job inhibiting the virus when we get to Beta and Delta and especially Omicron. We also looked to see whether there were differences in these correlations over the different immunological subgroups. And when we look at healthy volunteers versus people with any immune disorder as a whole, there is a statistical difference that the correlation is actually lower in people with an immune disorder than it is in the healthy volunteers. So even though you have antibodies, you may not be inhibiting as well. This varies by immune response, or excuse me, by immune disorder, as you can see in this table here. And so we really want to start exploring this in more detail to see why is it that some groups, like those with immune dysregulations, may have a higher correlation, may have their antibodies are able to inhibit more virus than the healthy volunteers, and why some groups, such as the combined immunodeficiencies, are less able to inhibit the virus. So this is something else that we're going to be looking into as we continue on with our study. The last thing that I want to cover are the breakthrough infections. So we did see a number of breakthrough infections in our study. We had 28 people who have immune disorders get a breakthrough infection, and 9 of our healthy volunteers. This was technically not statistically different, but you do see that more of our healthy volunteers, a higher proportion, seem to get breakthrough infections. When we look at the time to infection, since the last dose of the vaccine, it took about 4.5 months for people with immune disorders to get their breakthrough infection, and about 5.5 months for those people with for the healthy volunteers. When we look at the number of doses people had received before their breakthrough, 32% had received only 2 doses, 65% of people had received 3 doses, and 3% of people had received 4 doses. But if you see in this figure, the majority of our infections were during Omicron. And as I showed you in the previous figure, the vaccines are just less effective at inhibiting Omicron. And so it's not surprising that even though people got 3 vaccines, those vaccines just aren't doing as good of a job as blocking that virus. So it's not really surprising. It's going to be really interesting once we get data from these new booster vaccines that have the bivalent formulations in them to see what happens with breakthrough infections. So we are still recruiting. You need to have received fewer than 5 vaccine doses in order to be eligible to enroll. So we are only following people through 5 doses of the vaccine, because otherwise we will be doing this study forever. And while I like job security, we can't do that. So if you have not yet gotten 5 doses of the vaccine and you're interested in participating, you have to be willing to submit blood samples and fill out a few surveys for us. And you can either have an immune disorder or be on immunosuppressants, or if you don't have any immune suppression that you know of, you can be a healthy volunteer. Family members of folks with immune disorders, we love to have you enroll. So if you are interested, you can go ahead and email us. I have the email at the bottom of the slide here. And I just want to say a quick thank you to our whole team. This study has really taken a village. It's a huge study. It's an ongoing study. So many people have really helped out. And most of all, our study participants, because they have submitted so many samples. They give us so much information. They're constantly in contact. We really would not be able to do this without them. So with that, I will take questions, and please feel free to contact me if you have any questions about the study, about study design. I'm happy to answer any of those. So thanks so much.
Wow, that was fantastic. Thank you so much, Dr. Ricotta, for sharing the wonderful information, not just on the clinical trials, how that process works, but that incredibly valuable information on vaccine effectiveness. It's obviously at the forefront of almost everyone's mind here. So now we do have time available for Q&A, but before we begin, I want to remind everyone to remember that everyone's treatment and condition is unique, that the information presented during this session is not medical advice and is not intended to be a substitute for medical advice, diagnosis, or treatment. Please always seek the advice of your physician or other qualified healthcare provider with any questions concerning your specific medical condition. So I do see here in the chat, we have at least one question here, and it looks like it deals with perhaps your last point there. So the question we have, it's an individual who actually resides in Canada, and they ask if they might qualify for any trials or consults with you. They apparently state that they have a complement factor that's low, and they apparently didn't respond well to other vaccines. And she didn't know if it might be possible to be seen at NIH, participate in a study, and if so, if there might be any additional costs.
So for our study, we can unfortunately not enroll international participants purely for logistical issues. So it goes back to what I said earlier. Sometimes that dictates who we can and cannot enroll. Now, there are other studies at NIH that are studying a variety of different things that have to do with COVID. We have a website you can search for NIH COVID studies, and it'll come up specifically for all the different studies that we have there. We do at NIH see individuals from all over the world to study a variety of different immune responses at NIAID specifically. And so if there's something that you are specifically interested in following up on, you can either check our website, you can check clinicaltrials.gov, or you can have your primary care provider refer you if they think there's a study that you would qualify for. So definitely check that out. We do, for many studies, bring in international participants. And like I said, we study such a wide variety of immune disorders and other conditions here. That's definitely worth checking out.
Great, thank you. We do have another one here in the queue. The question is, can you be in a study if you've had 4 vaccines plus Evusheld?
Yeah, so if you only had 4, so Evusheld does not count as a vaccination for our study because that's just giving you antibodies. It's not encouraging your body to produce its own. So yes, for our study, you would still qualify. If you're interested in looking at other COVID vaccine studies that are happening around the country or the world, you would have to check their inclusion criteria specifically. But for ours, yes, you would qualify.
Great, and there's a follow-up to that, and the answer may be sitting on the screen here in front of us. They've asked if you could share the email for the trial specifically.
Yep, that's
So
this email down here. It's NIAID COVID vaccine study at niaid.nih.gov. You can also search, we come up on the NIH COVID research website. We are also on clinicaltrials.com, clinicaltrials.gov, so you can search for us there as well.
Excellent. Thank you. I'm going to give folks a few more moments. We've got this wonderful expert and Dr. Ricotta sitting here, folks, just waiting to take any of your questions. So I'm going to give folks another few moments to get their thoughts together and perhaps do that. So I have a question for you while we might be waiting, Dr. Ricotta.
Sure.
So I saw the graphs and the charts looking at the response of the vaccines to the various variants, and we know we're always going to be chasing the latest variants. Do we know if there are any specific levels that are protective against these? So for example, even though if the response might be lower, is there still at least some protection or cross-reactivity to these?
So there's no magical cutoff value that we're aware of. It really differs by individual. Also remember that antibodies are not the only part of the immune system that's going to protect people from COVID infection. We also have the T cell response. And so it's really hard to say whether, oh, I've produced X number of antibodies, I'm protected now. We don't say that, we don't recommend that. This is why we don't, we're kind of general when we do share these results. But it does look like, as I showed in the figure with the bunch of the lines there, that was, it does look like the more antibodies that you produce, at least for earlier variants, the more protected you are. There is still cross-reactivity, even though that the inhibition level does drop down for Omicron, there is still some protection, some cross-reactivity. And again, we also hope that the vaccines do help induce a T cell response as well. And so that can also help protect against new variants of COVID that we see cropping up. There are vaccines that are currently in development that are trying to target areas that are less variant, that change around, have fewer mutations on the virus. So that would give us longer term protection against new variants that may arise that we don't know about yet. So those are in process. They're still in early phase trials. I have no idea when they might be available or how effective they will be once they hit the real world. But scientists are currently trying to do that, so we don't just have to keep chasing the virus like we do with flu basically every year.
Yeah, a universal coronavirus vaccine would be wonderful for everyone,
would be.
particularly the immune compromised. We do have another question here. So the question is, what is the best way to keep track of study updates, specifically your COVID study and also Dr. Del Monte's similar study for those with inborn errors of immunity?
Oh yeah, Dr. Del Monte and I work together. And so there are a couple different ways. Study updates do come, they are slow to arrive. We are really just working on our first study paper right now. But you can check through. Sometimes the NIAID Media Office will post study updates. You can follow them on Twitter, on Instagram. You can go to the NIAID Media website. I know I post study updates on my Twitter, which you can find there. So you can always check there. If you are enrolled in the study, I send out periodic study updates. So that's how my study participants follow through. But really, while we do try to make an effort to do presentations like this, so that the participants and interested folks in the community can find out about the study results, sometimes it does take us quite a while to make sure that we get the data we need, that it's well validated, that everything, we're really solid in our conclusions. So if you don't hear of anything from our studies for a little bit, don't despair. We will have updates that come out because we are really eager to get this information out to everybody.
Great, the questions are rolling in now. This is great. So thank you everyone for adding your questions. I'm going to get to as many as I can. We have a decent amount of time left, so keep them coming. So question here, where would one go to get the blood test associated with the study?
So for our study, we are actually using tests that we've developed at the NIH. They provide us with a lot more information than any of the commercial tests do. So we're actually able to count the number of antibodies that people have in a sample. Whereas the commercial tests don't tend to do that because they can be expensive, they're hard to interpret. If you are interested in finding out what your antibody response might have been, there are tests that are called semi-quantitative spike IgG antibody tests that you can have your home physician order. Quest Diagnostics does them, LabCorp does them. Most clinical labs at this point can do them. Now, the thing with these is that they are, again, semi-quantitative. So they are not going to tell you the exact amount of antibodies that you produce. They'll basically just give you a yes or no answer. So either you have antibodies or you don't. The hard part is that it's difficult to interpret what that means in terms of your level of protection. But if you want that peace of mind that you have any antibodies at all versus none, those tests are available to you. The tests that we run here are just research-based. We don't have FDA authorization to use them as clinical tests, and so we don't do them for individuals outside of the study.
Great, thank you. Another question. This individual states they have 2 genetic mutations, one of which is immune system related. The other is completely unrelated, yet does have a significant effect on their health. Would having both of these genetic variants prevent them from participating in the study?
Not necessarily. We evaluate everybody on a case-by-case basis. If whatever it is, the secondary mutation that you have, as long as it is not going to confound our understanding of your immune response to the vaccines, then it should not be a problem. But if you were to say you don't produce T cells because of your first mutation, and then you have to take a B cell suppressing medication for your second one, we may need to have a little more detailed conversation with our whole clinical team here to make sure that the results that we get from you, we can interpret, because we need people, the data that we get, we want to make sure that when we interpret it, we know what it means, so that we're not just coming out here and presenting and saying, well, we've got some data, but we have no idea. And so that's another reason why we're very careful in who we select for our studies, to make sure that we're able to actually understand the results. So to answer your specific case, we would have to talk more about what you have, talk with the clinical team, but it's not disqualifying right off the bat for sure.
Thank you. And you mentioned this, and this kind of leads into the next question. You've mentioned it twice in the last few moments, which is a question from someone asking if there are actual clinical trials or studies specifically looking at the T cell response to COVID vaccines.
So our study is going to be looking at the T cell response. T cells are harder to study than antibodies are, which is why actually for any of the clinical trials that Moderna, that Pfizer did, you don't see the T cell response as an endpoint because they're difficult to do. It's expensive to do it. We don't always know what that means because T cells are tricky. And so even the large clinical trials that were run for authorization of the vaccines only looked at antibodies as the endpoint, and that's why. So there are studies that are looking at T cell response to the vaccines specifically. We are evaluating that on our study. We're looking not at what's called a functional assay. So we're not going to look to see how well the T cells may actually be inhibiting viral replication or entry or things like that. But what we're looking to see is on your T cells, you have different receptors that are going to bind to COVID. And we want to see how many T cells you make that can bind to COVID and how many different parts on the virus they can bind to. Because the more T cells you have that can bind and the more diverse the population of proteins that it can bind to, the more likely you are to have better protection. So that's what we're going to be looking at. It's not a direct assessment of T cell function or how well the T cells are actually providing immunity, but it does give us a good proxy, and it is the first step in really getting a better understanding of this. There are other studies out there that have done T cell work. They tend to be smaller in number and scope, which is why it's still kind of a big question mark.
Thank you for that. There is a follow-up question about the labs and the blood work.
Mhm.
So if someone were to participate in the study, would they have to travel far to do the blood test, or is this something where the samples or the blood could be sent to you from a remote site?
So our study can enroll remotely. I specifically did not want to limit our study cohort to just folks in the greater Washington, D.C. area because I did not know how representative that might be for the broader immune deficient community. So you can participate remotely. There are two different ways that you can do this. The first is you can go to either local Quest Diagnostics. We have a contract with them all across the country. So you can either go to a Quest or you can go to a local clinic if you have somebody who's really good at drawing your blood or who does it regularly, and then they will mail the blood to us. If you do not want to do a blood draw, don't have a Quest close by, don't feel comfortable going out or going to a lab to draw the blood, you can submit a finger stick sample to us. So we send you a kit, you just prick your finger and put it on the samplers and you mail that back to us. That one provides a little, obviously less sample than a whole tube of blood. So we can't do quite as much with those samples, but we can still get a lot from that. Now I do want to highlight, because I know that this is something that continues to disappoint everybody. Like I said earlier, the tests that we are doing are not FDA authorized as clinical laboratory tests. And so I am not authorized to return a participant's individual immune response data to them. So even if you were to participate, I cannot tell you your level of antibodies because again, I don't know necessarily what that means in terms of your level of protection. So it would be terrible if I told you, oh, look, you've got all these antibodies and you assumed that you were protected against the virus and you went out and you got COVID and something terrible happened. That's why the FDA does not allow us to tell these results. And so if you participate, you are providing your samples so that we can look at this as a group level analysis, but you would not receive your individual level results. This is when we would recommend that you go and have, again, your local provider order one of those commercially available tests for you if you are curious. So that's just a caveat there to let everybody know so you're not disappointed if you decide to contact me.
That's a very good point. It kind of leads into the next question. An individual had a question about whether or not here, let's see, where is it? That they haven't had any COVID vaccines, but they did receive Evusheld. They wanted to know if they're indeed protected for 6 months or not, or anything like that. I think that kind of gets to the last point you made about returning results, but please go ahead.
Yeah, I mean, the hope would be that the Evusheld would stick around in your body for the 6-month time period that it is authorized for. It's also individual, though. There could be some reason that your body just doesn't, the antibodies in your body just don't persist as long as they normally would or should. And that would really be something so individualized that unfortunately, I can't give you a good answer for that. It's something, if you're worried, again, you can go and do one of these semi-quantitative tests to see if they pick up any antibodies at all, to see if you do have any sort of protection. But yeah, otherwise, unfortunately, there's just no way to tell.
Right, understood. So at the moment, we have one last question, and this is an interesting one, and I'm not sure how, if we can answer this, but the question is, the first part might be easier. What % of the US population do we believe have become infected with COVID? And what % of those with a primary deficiency do we believe have become infected with COVID?
So I don't know those numbers off the top of my head. They change so frequently. I have just not kept track. A large part of the population at this point has had COVID. Luckily, a lot of that has been asymptomatic or very mild illness, which is great. We have seen a bunch of severe disease. Once the hospitalizations start ticking up, we know we're back in a COVID surge. So in terms of the general population, a lot of COVID has happened. In terms of people with immune disorders, now it's interesting actually, because it doesn't seem like, again, it varies by the immune disorder, but many people that have gotten COVID, or even in our study or in general, luckily have had really mild illness as well. This could be due to the fact that your immune systems just can't ramp up enough to cause COVID disease, which is driven a lot by your immune system. And so if your immune system can't overreact, then you can't develop severe COVID, which is really good news. Now, you could potentially have viral persistence, which maybe is not such good news, but at least it seems like in many folks who have different primary immune disorders, it's not as devastating as we originally feared. Now, that's not the case for all. There are certain immune disorders that people do end up having severe COVID. Now, why is this the case? Well, we think that, a lot of you folks with primary immunodeficiencies, you know how to protect yourself from infectious diseases. Your behavior is different than people who are healthy. You don't, maybe you're still wearing your masks, or maybe you haven't gone out to eat indoor restaurants yet, or, you're skipping out on that concert that everybody else is going to. And that behavior change is actually potentially what's more likely to keep you from getting COVID than anything else. That's probably why we see a larger portion of our healthy volunteers. Remember, we saw about 35% of our healthy volunteers had gotten COVID compared to, I think it was 13% of our immune disorders. And that is very likely due to behavioral differences. That is something that we are actually currently analyzing. We have run a survey in our population to ask about behaviors before and after vaccination. But so it's not that necessarily the case that, everybody with an immune disorder has gone and gotten COVID or is going to get COVID. Those that non-pharmaceutical interventions that you still can practice and hopefully are still practicing, those go a long way to protect you from COVID as well. And so keeping those up, even despite variants and other upticks and all of these other things happening, wearing masks, keeping distance, all of those things can still go a long way to protect you from getting COVID. So that was a really long way to answer your question. I don't know the exact number of people with primary immune deficiencies who have gotten COVID, but at least in our study, it's fewer than the healthy volunteers.
No, that was excellent. Actually, at the very beginning of the pandemic, March, April, May, for the first few months, IDF ran a bunch of online surveys to kind of act as canary in the coal mine. And we found a lot of the same things, that thankfully, many of those in our community didn't seem to be any more impacted by the severity of the disease than non-immune deficient individuals. And it seems like other factors. So, here's a way to tell people that taking care of your health and other matters can help you with these things. So
Absolutely.
people who have obesity, uncontrolled diabetes, or hypertension are much more likely to have adverse or more severe outcomes from the COVID virus or the SARS-CoV-2 virus than those who had those things under control.
Mhm.
So an important point is take care of your health and other things as well, and any other conditions, and you're better able to weather a lot of these things. At least that seems to be the case with this virus.
Absolutely. It definitely is. I mean, being more healthy in general, being more protected in general is really going to help you holistically. We have noticed the last couple of years that flu has been down because people are wearing masks. And so they're not, that's helping protect against other respiratory viruses as well as COVID. And so really just having better prevention in general, better hygiene in general, and better health in general is going to help with not just COVID, but with lots of things. But it will also help with COVID. And so it's definitely best to keep doing what you're doing. This population specifically, you guys really know better than a lot of others how to keep yourselves protected. And that's a really good thing for you.
Well, I think that's the end of the questions. I'm going to give folks about 3 more seconds before we decide to close out here. This has really been fantastic. Again, I've learned so much. I've been in IDF since 2007, and every conference I still learn, and I've learned so much from you, Dr. Ricotta. But
Well, great.
questions. On that note, our time has come to an end. So again, thank you so much for your time. It's been an honor to be here to join you and have you with us today. So thank you from me and on behalf of all our community. To everyone out there, please enjoy the rest of the conference. There's still a ton of programming and things going on. Make sure you visit all of our sponsors and exhibitors and thank them. Make sure you hit all the networking sessions and enjoy the rest of your time at the IDF conference. Thanks, everyone. Have a good night.